in fMRI research, we have to deal with a lot of sources of noise which impact the signal that we're interested in negatively, particularly by decreasing the signal-to-noise ratio, or the functional signal-to-noise ratio, which the larger it gets, the better it is for our data. Those sources of noise include system and thermal noise, non-task related and cognitive variability that we find in our participants, cognitive and behavioral variability, that's also a problem, and movement and physiological noise. All of these aspects, all of these sources negatively impact the functional signal-to-noise ratio. Luckily, there are a number of things that we can do to counteract this, and those we'll discuss here now. They include cleaning up your data, increasing the magnetic field strength, and spatial and temporal averaging. So let's discuss the first aspect, cleaning up the data. So there are actually ways that we can obtain estimates of physiological noise directly at the level of the scanner. So the scanner has, for instance, an ECG. Most scanners have. So you can get an electrocardiogram by attaching electrodes to the heart. It also allows you to get respiratory information by using a breathing belt that can be attached to the participants in the scanner. And so you get this type of information, or possibly also via other sources. You can get arousal information from skin conductance responses, but also from the eye tracker that might be installed at the scanner. And this allows you to remove this noise from your data. And this is one way to do it, for instance, via the PhysIO toolbox, which allows you to get, directly assess the influence of the heart rate or heart rate variability on your functional images, but also of respiration on your functional images. So you can basically remove this noise and clean up your data in this noise modeling step here. That is part of, for instance, this toolbox, but there are other toolboxes that can accomplish the same or similar tools that are available out there. We also have the option of, if we haven't recorded this type of data, of using independent component analysis to identify noise in our data and then remove this noise by denoising as well afterwards. So independent component analysis will basically subdivide your data into separate components, obviously, as the title says, that are independent of each other, using, basically statistical, mathematical techniques to do so. And some of these components often are noise-related, purely noise-related components, related to, for instance, scanner noise or physiological artifacts. And this, once we identified those in our data, we can add them as regressors in our imaging models to remove these sources of noise. This is one way, another way to do it. So adding this to our data and removing these sources of noise that are common and we know of will improve our functional signal-to-noise ratio. Now let's think of another way. So if the primary determinant of functional SNR is the amount of net magnetization, I think it's quite easy to think of another way to increase functional signal-to-noise ratio by increasing exactly this measure, namely net magnetization. How can we do this? Well, the simplest way to do so is to crank up the magnetic field. And obviously that's something that we can do by, for instance, moving to a 70 scanner or a 90 scanner. We have one at our location here, but a lot of research groups also have access to 70 scanners. And typically using a 70 scanner is a little bit more expensive than the standard 3 or 1.5T scanners. And it turns out that a lot of research groups are paying extra for this. Well, why would this be the case? Well, because you do get better signal. Here is one example that shows the difference in signal between a 1.5T scanner, which used to be the standard, in the early 2000s. Now it's more closer to a 3T scanner and a 4T scanner. This is, again, a visual activation study where alternating blocks of flashing light and darkness every 30 seconds are presented to the participants. So you have these flashing checkerboards again, right? And you can see that that at the onset of a trial or of a block for 30 seconds, visual activity goes up, stays up, and then goes down towards the end. You can see this nicely in the 1.5T scanner, but the response is more than doubled in some cases here in the 4T scanner. So you can see that the effect of increasing field strength in this very specific case of visual activation is to increase and possibly even double the bold signal change, which is quite impressive here. Let's then have a look at what 7T can be good for. And this is one example we'll discuss here from the literature that demonstrates ocular dominance columns in the human brain using a 7T MRI scanner. To just give you a brief update about what ocular dominance columns are, it's basically a visual region or an organization of primary visual cortex, but specifically in cortical layer 4, that is organized into columns, which you may have encountered before when, for instance, when we talked about the organization of area MT, which is also a higher order visual region, into columns that code for specific directions. Well, here we're just talking about columns that preferentially respond to input from one eye versus the other eye. So this is a part of the visual system. Information from the retina, which is here subdivided into two parts, the temporal and the nasal retina, is distributed or is sent forward to the lateral geniculate nucleus, which then sends it to primary visual cortex. And what you can see here is that there are columns in primary visual cortex that tend to, so primary visual cortex is also divided into a left hemisphere and a right hemisphere, right? So let's say we're in the left hemisphere and we're getting left eye input. So this would be the left eye. Then this would be a column that responds preferentially to the left eye in the left hemisphere. So that's where there is no crossing over of the optic fibers, right? So we're actually talking about the same hemisphere eye sending information that's being processed in the same hemisphere. But there are also columns that care about contralateral visual input from the eye on the other side. So this being the right eye here, sending input also via the LGN, but a different subdivision of the LGN to a column that cares about contralateral input. And this is something we observe commonly in visual cortex, but also other regions, that there is this columnar organization of cortex into a certain type of neurons that preferentially process a certain type of stimulus. And then this is then adjacent to a different set of neurons that actually prefer the opposite or another type of stimulus. This was discovered in the 60s by Hubel and Wiese, who made electrophysiological recordings in cat primary visual cortex and found exactly this organization. But it has been pretty hard to establish the same findings in the human visual cortex until, well, there were studies before this one, but this one shows it really nicely using a specialized sequence. So this is the normal sequence that we know of, and this is a somewhat specialized sequence that seems to work best for these types of stimuli and to identify visual cortical ocular dominance columns. So what's done here is that there's alternating blocks of visual stimulation presented to different eyes. So to the left eye for let's say 40 to 50 seconds, and then to the right eye for 40 to 50 seconds. And then you can obviously quite easily observe in the visual cortex what regions respond preferentially to input from the left versus the right eye. So it's a very simple experiment. But given the low spatial resolution that we have, in relatively low or mid-field magnetic resonance scanners, MRI scanners. This has been hard to identify, but at 70, we can actually quite nicely, and you can see this, pick it up with your naked eye, see that there is an organization into left shown in blue, or contralateral shown in blue versus ipsilateral stimulation within the visual cortex, and you can identify this using both types of sequences for the bolt response. So maybe you have the highest spatial resolution in this type of image here, and then if you apply some smoothing, you can see this. So the advantages of 70 are clear from this experiment, because it allows you to identify even very small scale cortical organizations such as ocular dominance columns. And this is especially true that you increase the spatial resolution. You can see here it's about 1 millimeter, which is pretty high. We work with 3 millimeters, so it's a third of what we work with, allowing you to do this. This is especially true when you focus only on one part of the brain, because you can even scale down what you're looking at in the scanner. It also allows for faster acquisition. So it's always a trade-off that you're making between spatial resolution and faster acquisition, but you have to make less of a trade-off. It's still present, obviously, when you go to 7T. Now, one of the problems is this is not always useful. So if you're looking at these very specific anatomical organizations, it's very useful. But if you're interested in, let's say, ventromedial prefrontal cortex, which we are, then the use of high field MRI may not be so large. So this is showing a scan at 1.5T and this at 4T for 3 different subjects. And you can see that one thing that changes are these susceptibility artifacts. So these dropouts in the functional scans, so in the EPI scan that we acquire, they become much larger in higher fields. There are ways to counteract it, but they're never perfect. So this is one of the side effects that you have to deal with. You can see visual cortex is pretty much left untouched, which is why the previous study worked so well. But if you're interested in orbitofrontal cortex or ventromedial prefrontal cortex, then you have some disadvantages. So now let's go back to averaging, which is the third point that we can improve functional signal to noise ratio. And obviously we know that bolt measurements are composed of signal plus noise. And we make the assumptions here that signal is the same across repetitions and noise is random. Now we know already that's not true for all of the noise involved in acquiring functional images, but at least it is the case for most noise. For instance, scanner noise is not related. So then with sufficient repetitions, if we average the signals, then the noise will tend to average out, whereas the signal, which is the same across all of these repetitions, or at least similar, should be preserved. So we're getting to a point where even a weak signal can be detected through signal averaging, because the noise over many, many repetitions that randomly falls at different levels within each signal for each repetition will then be averaged out. So we don't need to know exactly how the noise looks like or where it comes from, but it's important that the noise is random and it's independent of the event of interest. And that's basically given by random noise. Now there are sources of noise that are synchronized to the event. For instance, let's go back to studies that use electrical shock. If you give electrical shocks at specific time points, let's say before a decision is made, then you have a problem where you enhance all of the physiological reactions, which are basically noise in response to a shock that occurs before a decision, via signal averaging. So you have a problem. But if you randomly distribute, let's say, these electrical shocks throughout the experiment to fall at different time points of the signal that you're interested in, then you can you can actually enhance the signal and you average out the noise. Just as an example here. But here's a clearer example of this, where signal averaging was done in the visual cortex by basically recording 150 to 200 trials for each subject 1 and 2 here. You don't need more subjects, but we need many instantiations of the same event. And simply a visual checkerboard was presented for 500 milliseconds. And then we draw randomly with replacement from that sample of 150 trials. And we end up with these different averages. You can see that the more you average, the less noisy this average signal gets, and the more overlap there is between the different averages. Here in the case of 25 trials, here for 36 trials. And for 36 trials, you see that there is this nice signal or bold response that we can extract from averages of just 36 trials. And the noise then basically has decreased relative to this original example. Finally, averaging also increases the spatial extent of activation, as you can see here. So this is the number of trials that were averaged for subject 1 and for subject 2. The more trials you average, the larger the spatial extent gets. So we don't just get better and cleaner bold signals or reflections of the bold signals, we also get a larger spatial extent of the activation through signal averaging. And even the topography of the activity changes as the number of trials increases. So this means, based on these results, you can see that the step from 16 to 36 trials is largest, and then the gain is a little bit smaller as you go higher than this. And here you can also see that the change from 25 to 36 trials also seems to be largest in terms of how much better the signal gets. So this has been implemented in designs. So we should repeat trials at least 30 times, but up to 50 times to basically be in this range of spatial extent of activation and to be able to extract the bold response from the signal. Less than 15 is likely highly problematic because you're not even in this range then where you have very little activation to something like a visual, one of these checkerboards that actually leads to very large activation. There's no hard threshold for this, however, and it's still an open question how to solve this problem, because sometimes we actually have experiments where it's not quite possible to get even to 30 trials of the same event. Those experiments are sometimes done. And then what we have to deal with is obviously a lower functional signal-to-noise ratio. Finally, just as a reminder, when we do smoothing, we also do signal averaging, but at the level of spatial signal averaging. So we're assuming that two voxels that are adjacent to each other have similar signal and random noise in each. And therefore, when we average the similar signal, which we do via smoothing, then we're doing a form of spatial averaging. We reduce the noise, at least the random sources of noise, and we identify the signal.